So our topic today is to discuss about probiotics in celiac disease and what is the evidence. And this topic was requested by our audience, and is a topic that's a question that it comes up many times during our clinical visits with our patients. So these are my disclosures. There is no relation with any of the probiotic companies that I have. And the objective of today's review is to address concepts on gut microbiota, probiotics in celiac disease, and to learn what type of research studies have been done to evaluate whether a probiotic is effective or not, and what are the current recommendations on when a probiotic can be used and what type of probiotics in celiac disease. So to begin with the first topic in the agenda, we need to define what is microbiota, right? So you may have heard this word many times. It's now like a common language. Microbiota are microorganisms, are bugs that inhabit the human body, right? And within all these microorganisms, we have bacteria, viruses, fungi, phages. So there are multiple microorganisms. There are more like millions and millions of microorganisms in the body. And as you will see, we have different types of microorganisms. Each of these colors are different type of microorganisms, bacteria, viruses, fungi, and there are different proportion or composition of these bugs in different parts of the body. In the gut, which is the area that is affected by celiac disease mainly, we have different composition of these microorganisms, and the concentration of these microorganisms are more in the colon. As you will see there in purple, there are more concentration of microorganisms, millions of them, and less in the small bowel, much less in the proximal bowel, which is where celiac affects, and the stomach very rarely. So within these microorganisms or microbiota, we have bacteria, as I said, and within these bacteria, there are bacteria that have more beneficial effect, which are commonly known as good bacteria. Not sure that always is exactly that, but it's beneficial effect. For example, lactobacillus, which is a very well-known probiotic bacteria. And also you will see bifidobacterium, Akkermansia, Faecalibacterium promesi, and Ruminococcus. And then you have some bacteria that may produce or may induce some inflammation. E. coli, which produces gastroenteritis. Clostridium difficile also produces a lot of diarrhea, anaphylactic colon. H. pylori, that mostly produce inflammation in the stomach, mostly, and Staphylococcus, that is also producing as well multiple conditions in the gut and anywhere else. So these bacteria, good and bad bacteria, they live together and they should be in a proper balance. However, we don't have a very clear what is exactly a good balance, right? So that's why we cannot call them good, bad bacteria, good, bad microbiota, because there is, even though we know that there are good and bad bacteria that produce some benefits and harm, there is no clear what is a proper balance, and that changes within each individual. Maybe a good balance in you is not the same good balance in me, right? So these bacteria, this pool of microorganisms, they change since the person is born. Since as you will see, there are different concentrations that's shown in those circles, and then it changes throughout the lifespan. And why it changes? Well, because there are multiple factors that can change the concentration of these bacteria that can make it alive, or some of the bacteria or microbiota grow, and some others they come down, right? And what factors affect the gut microbiota? Well, diet, if the person does an exercise or not, if they are stressed or not, if they are taking antibiotics, probiotics, they change the balance of these bacteria and this microbiota. if they're taking medication, if they have different nutritional status, if someone is undernourished or overnourished. Also genetic, as you see there, since the baby is in the mom's womb, they have the genetic is already predisposing to gut bacteria. So these changes in gut microbiota in celiac happens, and we know that there is an altered gut microbiota that has been implicated as additional risk factor before celiac is developed, right? So there are some changes in this gut microbiota that has been implicated in the development of celiac that participates in the first immune reaction, but can also contribute to persistent on some of inflammatory status in celiac. So, and what bacteria has been known to change in celiac disease? Well, when different studies have studied like stool of people with celiac disease, and they have been shown that there is a pattern. Of course, no one has the same microbiota than the other, but there is a pattern with a trend to increase these bacteroid and proteobacteria and E. coli when the celiac is active, and decrease lactobacillus, bifidobacterium, which are known to be beneficial bacteria. And why this is important? Well, because the gut bacteria, each of these different types has specific role, and they participate in digestion of food, in synthesis of nutrients and some metabolites, in modulate the immune system, and also they maintain a healthy gut. What are the factors, as I mentioned before, that can affect the gut microbiota specifically in celiac disease? Well, if the celiac is active or if they're not active, they provide a different environment, and therefore the gut microbiota will change. Food composition, if people eat more fiber or less fiber, if they eat more fat or less fat, gut bacteria will change, gut microbiota will change. Also, in people when they are adults or children, as I show you in the picture, then it's important to know that when we establish the concentration of this microbiota, we do different testings. So the method that is used to measure this microbiota, it may change the results. In addition to that, where we take the sample, if we test the first part of the small bowel versus the middle part or versus the colon, and each side, they will change the concentration of different microorganisms. So it's quite, the reason why I'm bringing this is because studying this gut microbiota is quite complicated. A couple of years ago, this is a paper published led by Dr. Verdu, where we were testing gut microbiota in different sites and observing and confirming that in celiac disease, there are different gut microbiota in each of these specific parts of the duodenum. So when we do study, we have to be very, very specific where we're going to test. We know that in celiac disease, and to bring the celiac disease under good control, it requires a very strict adherence to the gluten-free diet. And in that way, the villus that were short when the celiac is active, they can grow back when the inflammation decreases and the regeneration of the gut the process take in place. However, it's well known that gluten-free diet is difficult to follow. Sometimes, people are struggling to adopt a gluten-free diet, even if they try the best. They are struggling still dining out, socializing, and also I hear all the time this major problem we're having that adopting a diet is difficult because it's very expensive, and because of that, well, Celiac Canada is working on trying to get more support for our patients. But because of all these issues, 30 to 40% of celiac had persistent symptoms and also shortening the villus despite trying their best to adopt a gluten-free diet. And because gluten-free diet is very helpful but insufficient because of this, and it's not an ideal treatment because of these challenges, there are novel therapies under investigation. And that's aiming to improve people's symptoms and to help, in addition to gluten-free diet, the person to become healthier and to feel better, and also to digest better the gluten. You can also, and hopefully in the future, some of these medications under investigation may help induce tolerance, but there is nothing yet there that with this purpose. But from all these therapies also, some of them, they're aiming to modulate this gut microbiota. And this is where the prebiotics, probiotics, postbiotics come from. So what are these biotics? Well, the prebiotics are mostly food that promote the growth of these good or beneficial bacteria. Probiotics are beneficial bacteria that can be grown in the gut. And these are the focus of our talk today. Then you will hear this word of postbiotics. We don't use that much, but there are components or metabolites or byproducts that this gut microbiota produces that could have an effect in the immune system, in digestion, in the gut. So in terms of probiotics, remember, and those beneficial bacteria, there are at least 1 out of 4 people with celiac disease will take supplements, and the most common are these probiotics. So in a large national survey that was done in the US, that was published a couple of years ago, 33 of people with celiac disease reported taking a probiotic. We are working now to establish how many people from our population in our clinic, and we're working with Hirday, are taking probiotics. So we'll have something similar, more numbers coming soon. Who takes probiotics? Well, mostly are people with celiac disease that are diagnosed later in life, and they have persistent symptoms despite adopting a gluten-free diet. Those people were more likely to take a probiotic. What are probiotics? Are live microorganisms that when you administer in proper amounts, they have a health benefit to the host, which is the person. There are different names that you will hear as probiotics. There usually comes in mixture of them, like combination of these bacteria, like bifidobacterium, Saccharomyces, Streptococcus, Lactobacillus, and some specific Escherichias that you will hear and you will see when you get a probiotic. Every single probiotic has different concentration and combination of these beneficial bacteria. And that's important because not all probiotics are the same. Every single of these microorganisms has different mechanisms. And what are examples of mechanisms? How they can improve health? Well, they can impact the growth of other microorganisms. For example, by growing good bacteria, they can impact on decreasing the growth of the bad bacteria. They can help digesting some gluten, not everyone, some of them. They can help digesting proteins. They have a proteolytic activity. They can help changes in gut permeability. They can help modulate the immune response. They can help with producing metabolite. For example, some of these probiotics may digest food and help produce metabolite, for example, histamine, methanes that can have a good or bad effect depending on how it's what the metabolite is. So they have different effects, like non-specific, that growth of different bacteria is non-specific effect. They have more species specific, for example, synthesis of vitamins, reinforcement of the gut barrier, that's specific for some species, and there are strain specific within the species. Some specific bacteria can produce different byproducts like inflammatory markers, can modulate the immune system. So they can be mechanisms from broad, like changing gut microbiota, to very, very specific on one single product that they affect. And how do you know how these bacteria acts and what's the benefit and what's the combination? Well, for that, you have to look into the research. You can conduct research, right? And there are different types of research studies investigating probiotics. You can go from preclinical studies, or those are basic research, and then you have type of studies in vitro. That's in the petri dish that you grow the probiotic and see what affects that probiotic. You can do studies in vivo, so you can put that probiotic into a mouse or another animal and see the effect on that mouse. You can do then clinical study, that's in the human population. And then from those clinical spectrum studies, you have different observational studies that you are not doing interventions. So people who are taking probiotics, you can see and do different tests, how they behave. Or you can do randomized trials, you can do clinical trials, and you provide the probiotic for the study. So there are different types of study. For instance, those preclinical studies, there are some studies in vitro, multiple studies evaluating probiotics like lactobacillus, BCL3, and seeing specific effects. Some of these studies in vitro are to change this concentration of bacteria, of the microorganisms in food, right? So then you can have a beneficial effect in yogurts, in bread, like you can have this effect. In mouse studies, there are multiple studies, and there has been multiple of these studies conducted at the Farm Institute by Dr. Verdú, Dr. Caminero, and I brought some examples of them, seeing the beneficial effect of, for example, this specific, the longum, bifidobacterium longum, or the lactobacillus that degrades some of these components that are in wheat, and that can decrease inflammation and can impact this digestion of these byproducts, can impact inflammation. Then there are other studies as well by Dr. Verdú's group that bacteria-derived metabolites, that those specific byproducts of bacteria that can reduce inflammation despite in mouse models of gluten sensitivity. So those are very interesting studies. And these are early findings. And when you see these effects in mouse, it's okay, that's fantastic. Let's start and seeing, because that effect could possibly affect a person, but you don't know exactly when you're doing a mouse study if you're going to have this same effect. And that's why you need then to do, after you've seen a very good effect in mouse studies, you have to conduct these clinical trials. So when we have a scientific evidence in probiotics, I want to give you background as doing clinical studies. Not all the studies provide the same level of confidence based on the evidence. For instance, I'm having you here this pyramid. At the bottom of this pyramid, the lowest level of confidence I will have in a study or in a statement on probiotic is an expert opinion. If a person is saying something or writing something in a blog, that's the lowest scientific level of evidence. Then we have case reports, and that happens. Case reports, and when we have one case that has a specific effect, we say, okay, that's interesting, and let's look into that. Case series, and we will have few cases that behave the same way. So if related to probiotics or any of these treatments, or then we have observational studies when there is no intervention, but we're observing people that are treated with probiotics. And then we have a systematic review of observational studies. What is a systematic review? Is a review in which is conducted in a very rigorous way, where a group of people are retrieving the whole literature. They draft first a protocol. They say, okay, we're going to look into these studies that meet these criteria. And then they're going to review everything that was published from the beginning until now. They're going to read every single study. They're going to see if they meet the criteria. They're going to put all the studies together, and then they're going to see the results. So it's a combined data from all the studies. That's why it's less biased than selecting the studies. Then you can do randomized control trials. That means that you are having these clinical trials and you're prescribing a probiotic and compare with something else, and the participant has the same chance to go one or the other, and that's why it's less biased. And then you have a systematic review of this randomized trial. That's the highest level of evidence that you can have. When you have a systematic review of randomized trial, that's a top level of evidence that you can have for scientific research. So then you have umbrella when you are reviewing all the systematic reviews, and then you have all the data that has been published ever. So of course that you are dependent on the quality of the study, but just to give you an idea what these terms that I'm going to talk about the evidence means. So when we talk about clinical trials on probiotic in celiac, on this randomized control trial, this randomized control trial is a type of study that is used to test whether a treatment actually works. If you're going to do a study, you want to ask the question, does this work? You have to do a randomized control trial. It is considered the gold standard in medical research because it reduces the bias by assigning by a flip of a coin, like different methods, and the person has the same chance to get one or the other treatment. So participants often get this treatment, for example, like a probiotic, or a control. That could be a placebo, or it could be a standard of care, or it could be another probiotic, right? So you're comparing two different treatments. The best evidence that you can have that something works is when you do the treatment versus the placebo, because you are reducing this effect that people improve because they believe in the treatment, right? So when people receive placebo, they don't know that they are receiving the placebo, which is no treatment. Then, and if they don't have an effect, that's a real scenario. And if they do have an effect, that's a real scenario, right? So that's why you have to control with this type of treatment. And there is we have conducted, now that you know the term, a systematic review, remember, pulling all the data, of randomized controlled trials, remember the top level of evidence for clinical trials to prove that something works, in probiotics in celiac disease. We did that in 2020, that was published. At that moment, there were 8 studies, randomized controlled trial evaluating probiotics in celiac disease, 4 of them in pediatric, 4 of them in adults. And what we found is that there was differences in study design, population, the type of probiotic used was different. There are the outcomes, so which mean that the effect that they were seeing, that were measuring, were different. So it's very difficult to combine and to put all together when the studies are so different. But what we were looking with this systematic review is to see whether the probiotic will improve symptoms in people with celiac disease, with improved quality of life, with decreased inflammation, with improved the gut lining, will improve these blood markers, and will impact and produce changes in gut microbiota. You know all these concepts now. So what we found, well, this is a typical graphic of a systematic review with the meta-analysis, with pulling all the data. What we found is the effect that you can see is based on that diamond. If the diamond touches the line, basically there is no much effect. You cannot have very much confidence. So that diamond is touching the line. So you have only one study that is on the left side that favors probiotic, and then the other studies is like there is not much difference. The other test that we do in this review is we look at the quality of the studies. If you see all green, then it means that it's a good study, good quality, no issues identified. If you see the reds, there are areas that we identify some issues. If you see yellow, we have concerns, but we're not sure. So the only problem we have here with that impacts the confidence is that all those studies were heterogeneous. That means that there were differences in all these outcomes that I mentioned before. But one of the issues that combining all of them is because they tested the effect of probiotic on symptoms using different questionnaires. And when you use different questionnaires, then you cannot combine this. You cannot compare these studies. But when we separated the studies by questionnaire, the ones that use as GSRS is one of the questionnaires that we use in studies, you'll see a good effect of a probiotic, a very clear diamond on the left in favor of probiotics in improving symptoms in celiac disease. And the heterogeneity, when we divide them and separate it by questionnaire, decreases significantly. So what we conclude from this is that probiotics may be effective. When we say maybe, it's because there are some red dots there. So, okay, we're some bias that could be in some of the studies, but they may be effective as in adjuvant to the gluten-free diet to improve symptoms in celiac. However, there were very limited number of studies that were evaluating this, right? And also the difference in study designs and all this that I mentioned may limit our confidence that this is really like true. So after this, that was 5 years ago, we decided to update. What is required when you conduct a systematic review is to every 5 years or so to update that. So thankfully, we have Hirday, a very, very talented student that he was working already in systematic reviews, and he kindly agreed to update this. He has been working with Griffin Carris, a medicine student as well, together and under my supervision to update this systematic review. So I will pass now this ball to Hirday to talk about what he has been doing and what he has been finding until now. Hirday.
Yes. Thank you so much, Dr. Pinto Sanchez. So yes, hi everyone, my name is Hirday. As Dr. Pinto Sanchez mentioned, I'm an undergraduate student working with her to basically update this past systematic review that we talked about and see what the new evidence says and see how many new studies are evaluating the effect of probiotics on celiac disease. So we started this project, I'd say, about in May last summer, so about a year now. So we registered the protocol on Prospero, so it's an international registry where you basically pre-register all your systematic review data on what you're going to do, how you're going to do it, just to ensure clarity in how a systematic review is done. And then we basically developed a search strategy. So a search strategy consists of many, many terms like celiac disease, probiotics, different terminology, and we applied that search strategy to 6 large academic databases, which gave us a pool of about 7,000 studies to sort through. And myself and Griffin, we went through these 7,000 studies to identify studies that were potentially eligible and could be included in our review as eligible papers, of which we found 17 papers that were eligible for the review, and 8 of these were actually in the old review, and 9 of them were actually new papers that we found that were published after 2020, which necessitated to basically update all of our estimates and see can we make any new estimates or can we make any new conclusions based on all the new papers that we've found since that past review was published. So if we go to the next slide, we can take a look at the 9 studies that we found. Now, while we did find 9 unique publications and papers out of our research, all these studies only really reported on about 6 original randomized control trials. So you can see here on the left that one of those RCTs was reported on by 4 different papers. So while we have a lot of studies, we don't really have that much original data. And we see that we run into a bit of the same issue again that Dr. Pinto Sanchez was talking about with the heterogeneity and the differences in all this data. We can see that 3 of these studies were in children and 3 of these studies were in adults, but a lot of them weren't actually placebo-controlled, and we weren't able to find data for even one of these studies. And a lot of them used various different combinations of probiotics, which range from normally a combination of either lactobacillus or bifidobacterium, but they did them for wildly different amounts of times. One of the studies was done for 6 months, while the other study was only done for a month, which really limits our ability to be able to say that, hey, probiotics do work or probiotics don't work when we are pooling all this data together into a meta-analysis, for instance. And so we had to drop these 2 studies that are highlighted in red since they did not have any placebo-controlled for the Elise study, and for the Otten study, we weren't able to find any data available. It was only a small abstract that didn't have the full data completed. And as you know, as we said, that a placebo-controlled trial is the best form of evidence, so we only chose to focus on those in our review. Now, if we go to the next slide, we can see our total chart of all the included studies that we included in our review, and all the ones that in red there are from the past paper, and all the ones on the right there are from a new paper in gray. And now we can see a lot of differences in these studies, like I mentioned previously. A lot of them don't assess symptoms. A lot of them assess symptoms on different scales. So some use the GSRS, some use the CDQ, which is a completely different questionnaire. So it really limits our ability to be able to pool all these studies together and draw conclusions from these studies. Now, if we go to the next slide, we can take a look at the methodological appraisal of these studies. Now, if you remember back to what we were just talking about, the more red and yellow you see in the study is the more we are unsure or highly uncertain about the risk of bias in these studies. Now, what is the risk of bias? So essentially, bias can come from anywhere. A lot of the studies that we saw didn't fully clearly report all their outcomes, meaning they may have saw some events or may have saw some outcomes in their study that they haven't fully reported in the paper or in the trial, which can lead to the skewing of the results. So if they found something that was not super beneficial, they may report that it was beneficial, which leads to a higher risk of bias score on this chart. And now we can see that we have a large proportion of yellow and red compared to green, and majority of that does come from that selective reporting that I was just mentioning, which basically shows us and highlights the need for more clear and more large, rigorous RCTs in the study, since our study is definitely limited by the studies that we are including here. However, nevertheless, we do have one study that was completely low risk of bias. And now if we move forward to our meta-analysis, Dr. Sanchez, we can go next. So first we looked at the quality of life for one of our outcomes, and we pulled together all the data for the studies evaluating quality of life. Again, here we can see some differences. Two of the studies use IBS quality of life, the scale, and the other study used the CDQ. So we pulled all these together and adjusted for the different directions of scores, since on one of the questionnaires, lower scores meant higher quality of life, and for the other one, higher scores meant higher quality of life. And we conducted two different analyses to basically see how well probiotics changed in quality of life. Now, if we look back to the diamond that we were explaining a little bit earlier, we can see that it's touching the line for both of these graphs here, for both of the analysis, so that we can see that there's no real change in quality of life with probiotics when compared to individuals who were taking a placebo. So still relatively uncertain on how it could benefit patients in their perceived quality of life. Now, if we go to the next slide, we can take a look at the probiotics inducing changes in the microbiota. So we evaluated two main microbiota compositions. So we evaluated the change in Bifidobacterium species in the patients, and also any change in Lactobacillus species in the patients. Now, we mainly chose this because a lot of the probiotics that we mentioned were some mixture of Bifidobacterium and Lactobacillus or something of the similar. And what we found was actually pretty interesting, that we did see a significant change in Bifidobacterium species. So you can see that diamond is to the right there, so it's favoring probiotics. While meanwhile, in the Lactobacillus species, it's slightly to the right, but it's still touching that line there. So we couldn't conclude that we had any change in Lactobacillus species. So that's a relatively interesting finding, that we have a difference in one species but not in the other, which may influence how patients are choosing probiotics in the future, if we are able to have more clear and more concrete data in the future from more rigorous studies. Now, what's actually really interesting, if we zoom next into the specific studies, we can go to the next slide. If we zoom into the specific studies, even though we didn't find a pooled change in difference in Lactobacillus species, we can see that on a study level, there did seem to be some change in Lactobacillus as well. So as we can see here in this study in celiac children, while they had a no difference in Lactobacillus studies when we included them in the main analysis, we do see here that the study reported that there was a relative abundance change in Lactobacillus over time, while it remained unchanged in the placebo. And similarly, there was another study, if we can click next here. There was another study that also evaluated the same exact thing, finding that the change in Lactobacillus was higher in probiotics groups versus placebo groups. So this basically kind of shows us that while individual studies may report changes in Bifidobacterium and changes in Lactobacillus species increasing and report this beneficial effect of probiotics in patients with celiac disease, that the actual pooled estimates may differ due to differences in the study population, in the patient's ages, even the patient's histories, since some of these patients may not have been on a strict gluten-free diet, as that's not something that was clearly reported by many studies. So ultimately, other factors, including population, the probiotics used, and many other factors that we're not able to fully capture in our analysis, may impact how probiotics are shaping the microbiota changes and also symptom adherence as well. And if we go to the next slide, we can take a look at the other two symptoms that we evaluated. So we also evaluated two different studies, evaluated the immune changes in celiac disease. So tumor necrosis factor A is essentially an inflammation marker. So it's showing what is the level of inflammation in in studies with that use probiotics for celiac disease. And now we can't really conclude that we had a difference in probiotics for celiac disease since the diamond is still touching the line. But we do see that there seems to be some suggestive improvement in inflammation. However, we're still very uncertain, especially due to the high risk of bias for the treatment allocation and selective reporting of outcomes, which indicates that there were some issues that we identified in how the studies were ran and how the studies were set up. And if we go next, we can also see, so we also evaluated the celiac disease in serology and intestinal permeability as well. We found no changes in TTG, which is the blood marker for celiac disease. And we also found no differences in like gut health and gut permeability as well. So no real differences between probiotics and placebo in those, which we had 3 studies for TTG, so 1 pediatric on gluten-free diet, 1 untreated pediatric for celiac disease, and 1 untreated adult celiac disease. And no differences for intestinal permeability as well, which was for 1 RCT on untreated adult celiac disease. Now, all in all, when we see all these results and all these analyses together, we can see that there are a lot of differences in how the studies report their outcomes and what the studies are finding, which essentially point to how probiotics can differ. A lot of the probiotics, as we mentioned, are different, and they can have different effects depending on the study design, the population, and how the study is even set up. And now I'll pass it back over to Dr. Pinto Sanchez to talk a bit more on how probiotics actually work and what the mechanism of them is.
Thank you very much, Hirday. The goal of showing this data was to see how complicated it is to answer the question whether probiotic works or not, because it will depend, right? So you will see that works in some areas and some others it doesn't, right? So, and this is because different probiotics act in a different way. So these are the mechanisms, how probiotics work, where this is a little bit more complex as well than before. One of the studies that was published years ago that was done with a group of Argentina, we were able to dig very deep into the effect of one specific probiotic at the level of this was at the level of the gut, and see that this specific probiotic was producing a substance that decreased inflammation, right? That's why when you see a probiotic that works and you see the mechanism and you understand why and where is working, then you have more confidence that this probiotic will work in celiac disease. So like a newer study from Dr. Verdú and Dr. Caminero, that was shown also that other probiotics are working in the metabolism of tryptophan, right? So there's the metabolites that produce decreased inflammation are driven by some specific bacteria. So when you know which specific bacteria works in specific area and in this specific population, this is where you're more certain that this probiotic will work. So this is, and this table is showing, and I'm highlighting here, when we do a systematic review at the end, this is one of the most important tables, because this systematic review is what is guiding the guidelines. Based on this review, the guidelines will tell if people should take or not probiotics. And what this table, which is called gray table, is showing, where in terms of probiotic in symptoms, yes, as I say, it may work, but our confidence is low. We really need more like a confidence with better design studies and larger studies with more people, right? And whether probiotics improve diarrhea, very low. It may improve, but we are very low certain that could happen. Whether probiotics can change quality of life, with here the show, it may change very little. So people who are taking probiotics versus people who are not taking probiotics may have a very little experience and a very little change in quality of life. And the good point and the important aspect here is that none of the studies showed any side effects, like zero side effects coming from probiotics. And this is good because sometimes when we try a treatment, we say, okay, if there are no side effects, zero side effects, but there could be a benefit, sometimes we try, right? So whether probiotics can improve symptoms in celiac disease, but we really need to continue working on this and what specific probiotics can work better in celiac disease and what are the mechanisms. So just to summarize in terms of the safety, as I mentioned before, there were no concerns and they had similar side effects if people were taking probiotic versus placebo. And however, this is very, very, very important to ensure that probiotics are gluten-free, right? Because there were some studies, one that was done by Columbia University. They tested 22 probiotics, different brands, and then they saw that half of these probiotics, they had some gluten, right? So again, it's important that if we are trying a probiotic, if we're trying to see if that helps people with celiac disease, improving their symptoms, at least we make sure that they are gluten-free and they are like a recognized brand. That's important. And so to summarize, what's the evidence of probiotics in celiac? Probiotics are not for everyone. And I have this question all the time. My patients ask me, should I take a probiotic? Well, do you have symptoms? No, I feel great. So why you will take a probiotic? Why you want to improve, right? You're doing great. Your celiac is good. You're doing gluten-free, right? So it's not everyone with celiac should take a probiotic. However, if you have symptoms, you're doing your gluten-free and you still have some symptoms, it may help, right? So again, it may work to try as discussed with your doctor. Very important. If you're going to try probiotics, discussing with your doctor, then make sure that they're gluten-free. However, you need to know that the evidence supporting probiotics yet is not very strong. So because most of these studies that we show you, there are a small number of people, and the studies are very different one another. So it's difficult to combine all of them to make it larger. And so what we still don't know, and this is the question I have all the time, is which probiotic? Well, we still don't know which probiotic is better because there are no studies comparing all these probiotics head to head, right? So we don't know if one probiotic is better than the other. What we need more studies is on specific types, as I show you, with specific mechanisms that we know that are going to improve, like, some aspect in celiac disease. So what we really need now, and we're working on that, is carefully design human studies to test specific probiotic guided by early research lab, that the probiotic that you tested in mice and it improves and you detect the mechanism, and then you test in humans, make sure that it's safe, and then you provide in the larger number of patients. Those are the studies that we really need to work on. So with that, I would like to thank everyone, and I'm very proud to work with people at work, and of course with all the nutrition initiative, the FarmCom. We're doing this series of webinars, and also we work together with Celiac Canada. And if you want to get into our McMaster Celiac Clinic website, you will see all the previous recordings. You will see all the work we have been doing, and you will see the research studies that we're doing. So the best way to get better evidence is by participating in research. So I encourage you to get more evidence on probiotics, is to participate in this type of research. And with that, I would like to thank everyone and for your attention. And I'm open for questions.